What is going on? It is Tuesday, March 8th, 2022. I had to pause for a second. I knew that was the date, but whatever. I was pausing to think of something else. Anyway, so I'm going to be talking about the rest of what follows glycolysis, specifically the aerobic pathway. Now, I didn't, from yesterday, if you are watching, remember, I didn't complete the detailed workings of glycolysis. I was going to go into the third step of glycolysis. I'm going to do that on another day, where I talk about the where I talk about what happens to fructose 6-phosphate and how it becomes fructose 1,6 bisphosphate. That is a whole entire process, requires a lot of regulation in that, and there's just so many processes that occur. What I have in the screen, actually, what I'm going to talk about, I'm going to be jumping around into a very specific section called the transition phase. So the transition phase is basically, it's what happens in the mitochondria if O2 is available. So remember, glycolysis can occur a certain way when oxygen is not available, and if oxygen is available, then that would be called the anaerobic pathway or fermentation. And if oxygen is available, then glycolysis would allow the electron transport chain in the mitochondria to be the end process that occurs, and the production of a whopping 34 ATP is the end result. So you get food, you eventually produce 34 ATP, and again, this is something that more complex organisms would have, eukaryotes would have, that have mitochondria would be able to do. Glycolysis occurs pretty much anywhere in any living organism, even the simplest ones, where 2 ATP, or most of the simplest ones, I think, that 2 ATP is the result, and that is a small result because that actually uses ATP in this process. Okay, so you got to give a little to get a little more, I guess. But anyway, and just a nice little quick detour is that if you want to get alcohol, right, the alcohol pathway, which is how people ferment wine, right, that organism goes through glycolysis and then has to go down this alcohol pathway, where the end result is CO2 and ethyl alcohol, and that's how you get all your favorite goodies that you like to go crazy talking over at night with your friends. Anyway, I drank a little too yesterday, don't tell anyone. But let's go on to the aerobic pathway, the transition phase. Okay, so the transition phase occurs essentially when when pyruvate, which is the end product of the first part of glycolysis, right? So pyruvic acid, aka pyruvate, is here, and as the glycolysis occurs, if O2 is possible, or O2 is available, then it goes into the aerobic pathway through the mitochondria, right? And so basically, let's talk about what happens in the transition phase. The end result of the transition phase, there are two major products of it. And the two major products are involved in the Krebs cycle. The major product that uses pyruvate is acetyl-CoA, right? And the other major products are NADH and FADH2. So those are needed for the Krebs cycle and electron transport chain, because they interact with the intermediates, and the Krebs cycle has a whole bunch of intermediates that need to be considered, right? Well, that's a future scoping or whatnot. But anyway, let's go back to the aerobic pathway, and here's your mitochondria, just kind of like how remember the aerobic pathway occurs in the mitochondria, so you can just remember where pyruvate eventually goes. So how does this occur? So this is actually fairly complex. But relative to all the other processes that we're talking about, it's not super complex. In the transition phase, where you get acetyl-CoA, you get essentially, and again, you get NADH plus, you get NADH, and you get FADH2, which is pictured here. You have an enzyme called pyruvate dehydrogenase involved. Okay, and so you have a enzyme complex called pyruvate dehydrogenase complex, which consists of three intermediate enzymes, which step by step go through creating the intermediate products until you get to the acetyl-CoA and the FADH and NADH, FADH2 and NADH. So what we start off with is pyruvate, right? Pyruvate goes in, CO2 comes out, and that's through the action of this pyruvate dehydrogenase. CO2 is a waste product, right? Typical, everyone knows that CO2 is the waste product. It's basically a carbon leaving the pyruvate molecule, okay? Because pyruvate, remember, is a 3-carbon molecule. I guess it might be pictured here or somewhere. Pyruvate's a 3-carbon molecule, and it gets converted into a 2-carbon molecule, and that extra carbon leaves in the presence of oxygen. That's why oxygen is here as CO2, right? Then through the action of this vitamin called TPP, thiamine pyrophosphate. Thiamine pyrophosphate will essentially work with this pyruvate dehydrogenase to strip off this carbon, right? And it will create, as an end result, an acetyl group, okay? So, and that TPP, thiamine pyrophosphate, will become hydroxyethyl hydroxyethyl thiamine pyrophosphate, right? As it's essentially its action has taken. The action we'll go into in specifics on the molecular level later. It's quite involved, as all of these are. But the acetyl group basically goes into the next enzyme, which has this really god-awful name called dihydroxylipyl transacetylase, or as I've seen it before, dihydroxylipoamide acetyltransferase. Okay, wow. Okay, great. That's a lot of letters and consonants and syllables. But dihydroxylipoamide acetyltransferase is an interesting enzyme, as in that it uses to do its work. And again, well, let's just get a reminder of what its work is. You have CoA, right? And CoA is bonded to this sulfur, right? And CoA then, through this dihydroxylipoamide transacetylase, will essentially, the sulfur bonds will be involved in stealing hydrogen from this acetyl-CoA, from this CoA, and from the environment, and using this acetyl-lipoamide, right? And you have that kind of attaching the acetyl group to the CoA group. Okay, so again, the mechanism we can go into later. There's a lot involved there, but it's called an acetyltransferase because it's transferring the acetyl group and transferring hydrogens off and combining it with the CoA. And so acetyl-CoA at this point is produced, and then that product is released and responsible for is available for the Krebs cycle and electron transport chain. But then we also need the FAD and FADH and the NADH. The FADH2 and the NADH are also needed, right? So these are then initiated. The production of these are initiated through the third and final enzyme in this pyruvate dehydrogenase complex called another mouthful of a name, dihydroxylipoamide dehydrogenase. Basically, and what this does is that this adds hydrogen, right? Dehydrogenase, that's what dehydrogenase enzymes do. They add hydrogen to NAD to create NADH plus, and then you have add hydrogen to FAD, or add 2 hydrogens to create FADH2, and then that is finally what happens and gets sent over to the rest of the downstream process. Okay, so that's quite a bit that happens there. And going into a bit of the mechanisms for these, it's actually quite interesting. Like, let's see if we can have a good crystal structure, precursor crystal structure. We eventually want to go into the details here, but yeah, obviously you can look all that up on Wikipedia. But the molecular breakdown is essentially again, we can break these down into the different sections. You have this dihydroxyethyl, this TPP, involved in the first pyruvate dehydrogenase. Oh, and also one thing you might want to just keep in mind is pyruvate dehydrogenase. What it's actually doing is performing a carboxylation reaction. So that, right, because it's removing a carbon and creating CO2 when oxygen is present. But essentially the end result is, I mean, the name looks a little weird, and I have to go into more of why that is besides the hydrogen activity that occurs as a result. So it's essentially a dehydrogenase that's also doing decarboxylation. But anyway, but just to kind of look at the structure, as we see it, dihydroxyethyl TPP, hydroxyethyl TPP goes between lipoamide and then finally goes to acetyl dihydrolipoamide, right? And those then are essentially the intermediates of what's occurring in the secondary process, right? So yeah, that's it for now. We'll go into the active sites and all the other details at some other point. Again, I'm going over all of these enzymes one by one, as I went over glucose 6 isomerase yesterday, glucophosphate isomerase yesterday, and the mechanism and the active site. I'm going to go into the mechanisms and active sites and the amino acids involved and the chemistry involved in each of these enzymes later on. So thanks for my little rant, and like, subscribe, etc., and I'll see you around.